All right, guys, so this video is to discuss a little bit about neuron physiology. So it is section 12.4 in the textbook, but I find that it's something that, when I first was learning anatomy, I had a hard time grasping. So I thought I would make a little video about it to explain it to you. So before we can talk about how a neuron fires and how the signal is passed along the length of a neuron, we have to establish what we'd like to call the resting membrane potential. Now, let me get my pen turned on here. Okay, so the resting membrane potential okay, for the rest of this, I will abbreviate that as RMP. Oops, not R. RMP. Okay, so the resting membrane potential is describing the differences in the charges on either side of the membrane. Okay, so essentially what it means is how much more negative is one side of the membrane than the other. Okay, so at rest, your cells hold a resting membrane potential of about negative 70 millivolts. Okay, and what that means is the inside of the cell, the intracellular fluid, okay, what we call the ICF, is 70 millivolts more negative than the outside of the cell, or what we call the ECF, the extracellular fluid. Okay, so at rest, the inside of the cell is negative 70 millivolts less, really is 70 millivolts less in charge than the outside of the cell. Okay, so this is established mostly because of potassium. So when we look at this picture right here, let me establish what some of these colors are real quick, because that'll make things a little bit easier to see. Okay, so the red circles here, let's do this one right here. The red circles here, this is sodium. Okay, and I do plus because it has lost an ion, so it's a cation. So it's a little bit more positive because it's lost that negative electron. And then the red, I'm sorry, not the red, the blue here is your potassium. Again, mostly a positive charge. Now you're probably looking at this going, if there's a bunch of positive ions inside the cell and a bunch of positive ions outside the cell, why in the world is it more negative inside the cell than outside? Okay, so most of that negativity has to do with these like orangey, goldish, yellow things, and those are proteins. Okay, these proteins help to, that's supposed to be an O. These proteins, whoa. These proteins help to give the inside of that, that cell a more negative charge, okay? So what happens is, and the reason that the cell is more positive outside than negative, because if the inside is negative, you would expect it to attract all the positive things, kind of like a magnet works. But what happens is this membrane here is very leaky to potassium. Okay, so potassium can go through the membrane pretty easily. Okay, and so what that does is it leaves the inside of the cell more negative because those positive potassium ions are leaving the cell. Okay, then what ends up happening though is that the because it is negative inside and there's a lot of positive things outside, right? The negativity of the inside of the cell is going to attract the positive charge of the potassium. So the potassium is going to leak through just because of its size and stuff. But then it's also going to be attracted back into the cell because of the intracellular fluid's negative charge. Okay, so that helps to kind of establish this membrane potential here. But what also happens is that the sodium, okay, again, sodium is a positive ion, excuse me, and it is also attracted to the negative charge on the inside of the cell. But we still need to maintain that negative on the inside and positive on the outside, because if sodium and potassium are all being attracted inside the cell, then the inside of the cell becomes positive. And when that becomes positive, then that's when your action potential starts. We'll talk about that in a minute. And you don't want your neurons firing all the time unless it's necessary. Okay, so what cells have is a sodium potassium pump. Okay, this pump is super duper important. It's probably one of the most important pumps in your body. It is the most energetically expensive pump in your body. From every time that the pump operates, it uses 1 ATP, and it operates a lot. Again, because the potassium is moving out of the cell, we have to establish and get that potassium back into the cell. Okay, so how this works, it helps to keep the concentrations maintained. So let's write that down. So it keeps the sodium and potassium concentrations. Again, these brackets mean concentration. Keeps it maintained. Okay, and how it does that is every time it operates, it pumps 3 sodium ions out of the cell and 2 potassium into the cell. Okay, so every time, great, and you can find some really good pictures of this online, and which I forgot to put on here. But every time it operates, it pushes 3 sodiums from inside the cell out, back out to the extracellular fluid, and it takes 2 of those potassium from the extracellular fluid and pumps it into the intracellular fluid. Okay, so again, this helps to maintain this negative 70-ish. It really runs between negative 60 and negative 90, but negative 70 is kind of the average. It maintains that negative 70 millivolt charge differential on either side of the membrane. Okay, and so what you end up with again is your more negative intracellular fluid and a more positive extracellular fluid. Okay, that is when a cell is at rest. It is not conducting any type of electricity going through it. Right now, when a signal reaches a cell, okay, there are 2 types of potentials that could happen. Okay, the first one we're going to talk about is the local potential. Okay, these local potentials occur when a neuron is stimulated by a ligand, right? And that ligand binds to a receptor. Okay, so kind of like it can be heat, it can be temperature, it can be a chemical signal like a ligand. So you have a receptor that is stimulated, that is stimulated by some kind of ligand. Again, the ligand is the molecule that sits in the receptor, and it turns that receptor on. Okay, and the receptor usually opens some kind of a channel so that molecules can move in and out of a cell. In this case, it's going to be the movement of sodium and potassium. Okay, so when the ligand binds to the receptor, it's going to open the channels. Okay, and sodium is going to flood into the cell. Okay, so your first step here is your ligand binds to receptor. Oops. Binds to the receptor. Okay. That receptor is going to cause the sodium gates to open. Now remember, sodium in a resting membrane is mostly outside of a cell. There's some inside, but most of it is outside. So when those gates open, that's going to cause sodium to go into the cell. So, okay, and because sodium is a positive ion, okay, which you can see here, it's going to cause the cell to go from negative 70, which is the normal resting potential. It's going to cause the potential of that cell membrane to go up. Okay, which means it's going to become less negative. So the inside of the cell is becoming less negative than the outside, if that makes sense, hopefully. Okay, so now as the sodium moves into the cell, your ICF, your intracellular fluid becomes, look if I spelled that right. So let me fix that real quick. Becomes more, it's an R, not a V. Okay, it's becoming more positive because those sodium positive ions are moving into the cell. Okay, what that is doing is it's making the membrane less polarized, okay, So what's happening here is a process called depolarization. And we're going to see that again here in just a minute. And in case you're at all unclear about where that term comes from, when you think of the term polar, think of having opposite charges on either side of a membrane or either end of a molecule. So when your resting membrane potential is the cell membrane being polarized, it means it's positive on one end and negative on the other. So when it becomes depolarized, you're getting to a point where the charges on either side of the membrane are becoming roughly the same. So that's what we mean by depolarization. Okay. And so what's cool about local potentials, okay, is that the local potential, the strength of it is directly correlated to the strength of the stimulus. Okay. Usually it's not super strong. It's not enough to stimulate an action potential, which is why it's called local. Okay. And because it's not strong enough to stimulate an action potential, okay, we can say that it is what we call decremental. Okay. So decremental means the signal is going to weaken as it spreads. Okay. So I'm going to go over all this stuff again at the very end after we talk about action potentials, and we'll kind of compare and contrast the two. Now, with local potentials, when the stimulus goes away, okay, the cell is going to immediately return back to that negative 70 millivolt resting potential. Okay, so it's what we call reversible. And then these local potentials can actually excite. So these potentials are exciting the cell, okay, whereas these here are inhibiting the cell. And what that means by inhibiting the cell is it means it's going to make it a little bit harder for that cell to become excited for an action potential. Okay, so these, what color have I got here? Okay, so these local potentials here, okay, we would call those excitable. Okay, good, again, good if I spelled that right. Hang on, let me fix this. Okay, and then the ones over here that are making that polarization more negative, we call those inhibitory, because they're making it a little bit harder for that action potential to take place. Okay, this particular one right here is one that is probably going to trigger an action potential, which we're going to talk about here right now. Okay, so local potentials are changes in the action, or I'm sorry, changes in the membrane potential that do not trigger an action potential. Okay, so there's a little bit of movement of sodium. Sorry, my dog's barking. A little bit of movement of sodium, but not enough to trigger an action potential. Okay, so let's talk about what an action potential is. Okay, let me go back to colors here. Actually, it's not that color. All right, so let's talk about an action potential. Okay, so an action potential, I like to think of as big changes to the membrane potential. Okay, and if I go back to the local potential here, this is really going to be small changes to the membrane potential. Okay, so now we're talking about big changes to that membrane potential. Okay, and what this means, or what this does, okay, is it occurs when there's a high enough concentration of voltage-gated ion channels in an area of a neuron. Okay, so if a stimulus occurs in an area or in a cell that does not have a high concentration of those voltage-gated channels, it's only going to cause a local potential. Okay, and in the book they give you numbers. I think a normal cell has 75 or 50 to 75 voltage gated channels per unit area, and then a cell that is that can trigger an action potential, they're going to have more I think it's up to like 200 in a, yeah, 250 to 5 or 350 to 500 of those channels per the same unit area. So this is going to occur in a nerve that's going to have a whole bunch of those voltage gated channels in one area. Okay, so here's what happens. Let me try to explain to you what's happening in this figure here. So your first step, okay, as numbered here, is your stimulus. Okay, so the stimulus is going to cause a current. That current is the local potential. Okay, so first step, we can say the current arrives at the axon Hillcock. So hopefully you remember from when you were looking at the structure of a neuron, it will look, I'm gonna draw this and I'll erase it. So it's going to look something like this. So my neuron drawing is terrible, but here is your cell body here. Okay, this comes down into an axon, which then goes to your axon terminals down here. So it's gonna kind of go like that. What in the world? Why is this happening? Hang on. There we go. So this is your cell body. Color that in so it all looks the same. And then off the cell body, you're gonna have all these dendrites. So the dendrites pick up the signals from the receptors, from other cells. And what it does is it funnels all of those signals into the cell body, which is that part right here. Okay. The axon Hillcock is where the cell body kind of merges with the axon, and the axon is going to be your long strip right here. Okay, so the axon Hillcock is this piece right here. Okay, so when that current arrives at the Hillcock, it really at that point is behaving like a local potential. Okay. So that's your Hillcock. Actually, I'm going to leave that picture there. Okay, if that local potential is strong enough, it will turn into an action potential. And what I mean by that is it has to get the voltage of the cell above, or that potential, the membrane potential above negative 55 millivolts. Okay, so again, resting is 70. So this is resting. Okay, it has to get it above 55. Negative 55 is the magic number here. Okay, these failed initiations that they're talking about here, those are just local potentials that weren't enough to trigger an action potential. Okay, so again, for an action potential to begin, that local potential has to change the voltage of the cell to negative 55 millivolts. Okay. So the local potential causes the I'm going to put this in a color because this is a really, really important number that you guys need to not forget. Negative 55 millivolts. Okay, so if it is less than negative 55 millivolts, then the cell or the nerve does not fire, as we call it. Okay, so that negative 55 millivolts is the magic number because a lot of those voltage gated channels will not trigger until negative 55 millivolts. If it's less than that, they stay closed. At negative 55 millivolts, they open. Okay, once those voltage gated channels open, then the sodium is going to rush into the cell. All right, so once that happens, once we meet that negative 55 millivolts, the neuron does what we call a fire. Oh my gosh, I really can't spell this morning. Okay, that neuron firing is going to be kind of, it's going to be the way that the signal from your hand, for example, that you put on a hot stove, that signal gets to your brain and back to your hands. So it's going to be a whole bunch of neurons firing in succession. Okay, when this happens, it comes in a couple different steps. So first thing that's going to happen, we've met that negative 55 millivolts. Okay, the sodium ion channels are going to open. Okay, and they're going to do this sort of slowly at first, but it's going to become faster and faster. So the amount of sodium that is going to move into the cell over time comes quickly. And when I say over time, if you look down here at the x-axis, this is milliseconds. So, you know, I say it takes longer. but really it's not that long of a stretch of time. Okay, as the sodium channels open, the sodium is going to rush into the cell, okay, which is going to cause the millivolts or the voltage of the inside of the cell to go up. So this is when the sodium ions rush in, as explained here. Okay, when that happens, again, we have that process of depolarization. Okay, the inside of the cell is becoming less negative than the outside of the cell. So let's write this down again. Depolarization. Okay. We like depolarization. We want depolarization to happen, especially if it's whatever that stimulus is going to harm our body. Okay. So the neuron has fired. The sodium is going, channels are going to open. The sodium is going to flood into the cell. This is going to cause the inside of the cell to become more positive, and the membrane potential is going to rise very quickly. That's why this line goes up so fast. Okay. Now, at some point, the membrane potential is going to pass 0 millivolts. And what that means at 0 millivolts, that means the inside of the cell and the outside of the cell are the same charge. The intracellular fluid and the extracellular fluid are the same charge. Once that happens, so at 0 millivolts, okay, those sodium channels are going to close. Okay, they're closed for business. Oh my gosh, they're closed for business once the potential meets or hits negative, or not negative, 0 millivolts. Okay, again, the charge is the same on the inside and the outside. Okay, so what's happening now, okay, as that goes up, once we're up here, the cell on the inside is now positive, so the intracellular fluid will become positive, and the extracellular fluid will be negative. This is exactly the opposite of the charges when the neuron was at rest. Okay, now we're in the middle of our action potential. Okay, as the voltage is going to peak, okay, your potassium channels are all open at this point. So the potassium channels will open kind of as that action potential gets stronger and stronger. Once we reach the top, the potassium channels are going to open. If I have enough room on this slide. Okay, those potassium channels are going to open. Remember, potassium at this point is at the highest concentration inside of the cell, right? And if you remember back from osmosis, things move from an area of high concentration to low concentration. So once those channels open, the potassium is going to rush out of the cell. Okay, it does that again because of the concentration, but the potassium, which is a positive ion, is repelled by the positive intracellular fluid. Okay, so it's going to rush out. Let's do rush to the extracellular fluid because it's repelled by the intracellular fluid. Okay, intracellular fluid is now positive. If you try to take two poles of a magnet that are both positive and put them together, they will not attach to each other. Okay, same kind of idea here. The positive molecules are going to leave the cell. This is going to, again, start to repolarize the cell membrane. Again, what that means is we're going to start to create a differential of charge on either side of that membrane. So this is repolarization. Right? Now, what you see here on this graph is a little bit of a dip in this area here. This hyperpolarization, this dip. Okay, this is caused by the potassium channel staying open a little bit longer than they should. Okay, so when all of those potassiums rush back into out to the extracellular fluid, the inside of the membrane is going to, or the inside of the cell is going to become more negative, or back to be more negative. So it's going to rush. It's going to hit the resting potential, and it's going to keep going again because the potassium channels are going to stay open a little bit too long. So it causes it to become super negative. Okay, that's when those sodium potassium pumps start to work a little bit of overtime, and it's going to push that membrane potential back up to that resting state of -70. Okay, that's what this dip is. That's that hyperpolarization because those potassium channels stay open a little bit too long. Okay, hold on. Let me go back. I need to add a slide I forgot to put in here. So let me put in a blank slide. Okay, and what I want to do is make a chart to compare and contrast the local potential to the action potential. Okay, so local potential. Ooh, that was not a straight line. I'm getting a little better with my little notepad, but the curve is steep. All right, so there's my straight line. And then action potential over here on the right. So action and local. Okay. Excuse me. All right, so in a local potential, okay, we say that it is graded. Okay, and again, what we mean by that is the strength of the stimulus, the strength of the potential is based on the stimulus. So if you have a super strong stimulus, that potential is going to be a little bit higher. If you got a super weak stimulus, that potential might be a little bit weak. Okay, so the strength is based on the stimulus. Okay, for an action potential, it is not graded. Okay, it is what we call an all or nothing potential or firing. Okay, so this one is all or nothing. Excuse me. Okay, if it is not enough to meet that negative 55 millivolts, the action potential will not happen. Okay, so it goes or it doesn't. When the action potential does happen, the strength of that action potential does not change with the strength of the stimulus. Okay, so again, it's all or nothing. Now, in terms of, let's put a line right there. Okay, in terms of how weak it gets, okay, remember I was describing the local potential, it goes to what we call decremental. Right. So the example they give in the book is like a ripple in a pond. So if you were to throw a rock in the pond, the ripples kind of get weaker as it gets further and further away from the point of contact. It's kind of the same thing here with the local potential. So it gets weaker as that potential gets farther away from where it originated. Okay. Action potentials are not decremental. So we can call those non-decremental. There it is. Okay. It is strong no matter how far it gets from the point of stimulus. It does not get weaker. And again, that's a good thing because you don't want the signal of the pain of your hand burning on the stove to get weaker as it gets to your brain, because then you're not going to be pulling your hand away as quickly if it gets weaker as it goes. All right, and then the last one, local potentials are reversible. Okay, so what this means is when the stimulus stops, so does the potential. And again, when I mean the potential stops, I mean it returns back to the resting negative 70. Okay, action potentials are not. Okay, once they start, they got to finish. Okay, that's what that means. Okay, now the last one, okay, we talked about the local potential can be either excitable or inhibitory to a cell. Okay. An action potential is always excitable. Okay. Okay. Again. Okay. Always excitable in an action potential, and then local can be excitable or inhibitory, as we saw in that graph I showed you a couple slides ago.